Alright, so class, I'm Cadet Matthew Clenathan. This is a presentation on isoleucine 5-monophosphate dehydrogenase. What is this enzyme? It's an enzyme found in bacteria, plants, and animals. It's specifically used in the synthesis of the glutamine. Nucleotides also used for just all purines. It controls the rate determining step in this pathway, and it's a key enzyme for DNA replication, which has its own effects. This is the overall mechanism that it wants to achieve in the cell. And this is a depiction of the quaternary structure. This is the eukaryotic version. Alright, here's the full reaction mechanism. The purpose of this enzyme is basically to get a hydride transfer and hydrolysis to convert IMP to XMP. What does that mean in the full scale? Is that you can start with a mixture of intermediates, and through this rate determining step, you can eventually get to guanine. I know this arrow points upwards, but eventually these XMP is a precursor to both guanine and the other purine. So here's a depiction of the primary and secondary structure. Here's the letters, and here's the depiction of the structure. So you see there's a lot of alpha helices in this enzyme. This one shows some beta strands depending on the species. This enzyme actually can differ from species to species, especially between prokaryotic and eukaryotic. However, alpha helices are definitely the common denominator of the two. The binding site, most importantly, has cysteine. It's called the cysteine 319 loop, and that's what binds the IMP to. About 503 residues in this depiction. There's other ones where it's about 498. It's a tetramer. The alpha helices and the turns tend to be outwards, and if you have beta strands, tend to be inwards with the hydrophobic part. We'll go to here. So what's unique about this enzyme is that it can, between species, have different dimers. So this would be a heterodimer. So the ligand that binds is potassium, and here it has 2 potassium binds, and over here you only have 1. And like I said, this is a heterodimer, but they also have a homodimer. So between the 4 subunits, you can also just have 1 potassium bind for each subunit. And this activates the enzyme to NAD plus and IMP. Its optimal pH is around 8. Studies show that you can use ammonium ion as a weak substitute for potassium. Sodium, kind of, but not so much, because it's very constricting with the strong nuclear force on the enzyme. It's hard to see this, but the cysteine 319 is the key step for when IMP and NAD will come in and bind, and then it has what's known as a flat. So eventually it will close. It will induce the reaction based on the hydride transfer and hydration, and then it will open up and it will release XMP, and it can keep NAD plus if it wants to. This is depicted in this. So it'll basically come in, and this is the NAD plus, this is the XMP, reaction will occur, and then it will release, and if it needs to, under certain biological conditions, it can close. Check that. Regulation is done by 2 nuclear factors for the gene that produces it, ATP2 and a palindromic octamer sequence binding protein. These allow general transcription factors to bind to the promoter region. It's seen heavily in T cells, and this is important and why people are studying it, because specifically human IMP-DH type 2 has been associated, especially with leukemia and some forms of cancer cells. Basically, because this is used in DNA replication, if they see human IMP-DH2 being created in large amounts, they know there's a tumor. So, and because it's a key enzyme for creating the purines, this is targeted specifically in chemotherapy. So here's a picture of some inhibitors. These these three are used in immunosuppressive chemotherapy for especially with leukemia patients, and this one's used in the antiviral chemotherapy, which is both these are synthetic compounds right now that are proved to be effective, but however the chemotherapy obviously pretty rough on the patient. Right now the big rush is to find some natural products, some found in nature, organic product that could competitively inhibit HIMT, VH type 2. And here are some candidates that some researchers have found so far. Any questions? No? Okay. Yes, Sam.
What practical research is going on?
So they're just trying to find natural competitive inhibitors, because that would take chemotherapy of the known compounds away. All right, as section marker, class dismissed.